In this video, we're going to be covering the following learning target. I can identify the location, reactants, product, and basic functions in anaerobic respiration. So last video, I discussed the fact that we have two different types of respiration. Aerobic cellular respiration involves and requires oxygen to be present in order for this process to happen. Anaerobic cellular respiration is a type of respiration that occurs when there is not enough oxygen or no oxygen around. Now, just to clarify something very important, before aerobic or anaerobic respiration takes place, glycolysis must and will take place. Okay, so glycolysis will always happen before either aerobic or anaerobic respiration takes place. Notice that aerobic cellular respiration produces a lot more ATP, while anaerobic cellular respiration produces much less ATP. So when there is no oxygen around, obviously the organism still needs to stay alive, right? Because when the body stops making ATP, when the body stops making energy, the organism will unfortunately die. To prevent that from happening, another process known as fermentation will take place when there isn't enough oxygen in the cells. Fermentation takes place in the cytoplasm. So just like glycolysis happens inside the cytoplasm, so does fermentation. Fermentation on its own as a process does not produce any additional ATPs. So you might be wondering, okay, then what's the point? Why does fermentation take place if no energy is being produced? The main purpose of fermentation is to allow glycolysis to continue to happen. And if you remember, glycolysis does produce small amounts of ATP on its own. Can you kind of go back to your memory and recall how many ATPs was produced by the process of glycolysis? So I'll give you a second to kind of think back to that. And the answer should have been 2, right? So glycolysis produces a net gain of 2 ATP molecules. So what's happening here is that there's a situation where there isn't enough oxygen, and instead of the organism dying because of no energy being produced, fermentation takes place, and fermentation allows glycolysis to happen. And at the end of glycolysis, we end up with 2 ATP molecules, which is better than nothing. There are 2 main types of fermentation that you need to be familiar with: lactic acid fermentation and alcoholic fermentation. We distinguish between these 2 types of fermentation by the products. Okay, so what is produced by the end of the type of fermentation. Notice that for both lactic acid fermentation and alcoholic fermentation, pyruvate is a reactant. So the reactants of both these types of fermentation are identical. Pyruvate is used, okay, and what's being made will depend on the type of fermentation. If we have lactic acid fermentation taking place, then of course lactic acid will be made. And if we have alcoholic fermentation being produced, then alcohol will be produced in addition to carbon dioxide gas. So alcoholic fermentation produces 2 things, or 2 molecules, while lactic acid fermentation only produces lactic acid. In addition, you will notice that this molecule known as NAD is being made. NAD is simply an energy carrying molecule, okay? This molecule, you don't really need to know too much about it in terms of details, but what you need to understand is NAD is an energy carrying molecule that will then be used in glycolysis to help glycolysis kind of take place, okay? Now, lactic acid fermentation, of course, is super useful for organisms that are trying to stay alive in low oxygen situations or environments, but it's also used day to day, by humans, and it has been used for hundreds of years to produce many of the things that we use and eat, such as soy sauce, such as bread and cheese and other dairy products. Basically, these products are created using fermentation. Notice that for all of them, we are starting with the glucose molecule that gets broken down into pyruvate, which this is happening during glycolysis. And then this pyruvate molecule will be used during either lactic acid, such as this type of fermentation for making soy or cheese, or alcohol fermentation, which can be used for bread and other, alcoholic beverages. So here I just have a general kind of review of the two types of fermentation. Again, we begin with glucose, which gets converted into pyruvate in both scenarios. And this is happening again during glycolysis. Glycolysis is the process that breaks down glucose into pyruvate. And then if lactic acid fermentation takes place, we will produce lactic acid. And this ATP, these two ATPs actually came from glycolysis, not the fermentation process itself. Same with alcoholic fermentation. We begin with glucose that gets turned into pyruvate during glycolysis, and then carbon dioxide, alcohol, and the two ATPs are produced. Remember that the two ATPs were produced in glycolysis and not in the fermentation process itself. So finally, let's compare and contrast aerobic cellular respiration and anaerobic cellular respiration. Right away, the main difference is that aerobic cellular respiration happens when there is large amounts of oxygen present, while anaerobic cellular respiration occurs when there's no oxygen present. Aerobic cellular respiration produces high amounts of ATP, 30 plus, while anaerobic cellular respiration does not produce ATP on its own. So fermentation will not produce any ATPs, but it allows glycolysis to take place, which produces two ATPs on its own. There are two types of anaerobic respiration: lactic acid fermentation and alcoholic fermentation. Now, the commonalities here, the things that are similar, is that they both are types of cellular respiration, and they both are changing glucose or converting glucose into ATP, which is a form of usable energy for the cells. In addition, in both aerobic and anaerobic cellular respiration, we are starting with a glucose molecule that is being broken down into pyruvate during the process of glycolysis. And in both scenarios, glycolysis is producing the net gain of two ATP molecules.